Dear students, good morning to all. Hope you all are staying safe and healthy. Today's our offline class is about goals of Human Genome Project HGP. Following are the important goals of HGP. Number first, identification of all the approximately 20,000 to 25,000 genes in human DNA. Number second, to determine the sequences of the 3 billion chemical base pairs that make up human DNA. Number third, to store this information in databases. Number four, to improve tools for data analysis. Number five, transfer related technologies to other sectors such as industries. Number second, Number sixth, LCELSI to solve any ethical, legal, and social issues. Number seventh, bioinformatics, that is close association of Human Genome Project HGP with the rapid development of a new area in biology. Number eighth, sequencing of model organisms. Non-human organisms' DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation. Many non-human model organisms such as bacteria, yeast, Canor habitus elegans, which is a free-living non-pathogenic nematode, Drosophila, plants like rice and Arabidopsis, etc. have been sequenced. As for example, in the table you can see organisms, base pairs, and number of genes. Organism E. coli with 4.7 million base pairs and 4,000 genes. Saccharomyces cerevisiae with 12 million base pairs and 6,000 genes. Canor habitus elegans with 97 million base pairs and 18,000 genes. Drosophila melanogaster with 180 million base pairs and 13,000 genes. Arabidopsis with 130 million base pairs and 25,000 genes. Oryza sativa 430 million base pairs and number of genes is 32,000 to 50,000. Now the methodologies. The methods involved two major approaches. Number one, ESTs, that is expressed sequence tags, and number second, sequence annotation. ESTs are expressed sequence tags identifying all genes that are expressed as RNA. Number second, sequence annotation, sequencing the whole set of genome that contained all the coding and non-coding sequences and later assigning different regions in the sequence with functions. For sequencing the total DNA from a cell is isolated and converted into random fragments of relatively smaller pieces. Recall, DNA is a very long polymer, and there are technical limitations in sequencing very long pieces of DNA and cloned in suitable host using specialized vectors. The cloning resulted into amplifications of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors were called as BAC, that is bacterial artificial chromosomes, and YAC, that is yeast artificial chromosomes. The fragments the fragments were sequenced during automated DNA sequencers that worked on the principle of a method developed by Frederick Sanger. Sanger is also credited for developing method for determination of amino acid sequence in proteins. These sequences were then arranged based on some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome was completed only in the sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, that is 22 autosomes and X and Y, to be sequenced. This is all about today's offline class. Thanks.